Welcome back, biologist. In this screencast, we'll finish Chapter 1. At the end of our last screencast, we talked about experiments. Once an experiment is completed, scientists will want to repeat their experiment to make sure that their findings were correct and to increase confidence in their scientific conclusions. When you conduct an experiment, sample size is also very important. Sample size is defined as the number of experimental subjects or the number of times an experiment is repeated. The larger the sample size, the more confident you can be in your results. Confidence in results can be measured mathematically by something called statistical significance. Again, statistical significance is a measure of confidence that results are real and not due to random chance. Let me give you an example. If you have a quarter, flip it 4 times, and get heads 4 times, how do we know if the results were due to chance or due to the use of a trick coin? We can measure statistical significance to give us an idea of the probability of this happening by random chance. After you do your experimentation, draw your conclusions and confirm them, you'll share your work in peer-reviewed journals. Peer-reviewed journals allow the scientific community to review and learn from your work. A hypothesis or proposed scientific explanation that continues to hold up after many years of rigorous testing may eventually become a scientific theory. Scientific theories are widely accepted amongst the scientific community. They're much more than just guesses. Be careful because the way we use the word theory in science is very different than the way we use the word in normal conversation. This infographic from the textbook describes the difference between everyday theories and scientific theories. Everyday theories are ideas or hunches. Scientific theories are scientific hypotheses that have been supported by hundreds or thousands of scientific experiments. After a great deal of experimentation, we have a large body of evidence about the effects of caffeine. This infographic shows many of the powerful side effects that caffeine can cause. It also shows that to minimize these side effects, the maximum recommended adult dose is 400 milligrams per day. Not all scientific questions can be answered through controlled experiments. Sometimes there are questions that will require us to go out into the field and make observations or comparisons to find patterns that will help us answer them. For example, if we want to understand the prevalence of heart disease or death by heart disease in different parts of the United States, we'll have to go out into the field and collect data. This data can then be used to produce maps like the one you see on this slide. Biologists who specifically study patterns of disease in populations are called epidemiologists. Epidemiologists, as you can imagine, have been pretty busy during the COVID-19 pandemic. An epidemiologist studying caffeine might ask, is there a relationship between coffee drinking and the incidence of Parkinson's disease? Parkinson's disease is a neurological disorder that affects muscle control. Epidemiologists will make observations and look for patterns that show a consistent relationship or link between 2 variables. If the 2 variables vary together, then we say they are correlated. We have to be careful with correlation. Correlation between 2 variables does not prove that one causes the other. Correlation is not causation. These graphs might help us understand correlation. When 2 variables increase together, we call that positive correlation. When one variable increases but the other decreases, we call that negative correlation. When there seems to be no pattern, we say there's no correlation. Just to reiterate, correlation is not causation. Let me give you a couple examples. Between 1991 and 2009, housing prices and the number of babies named Ava both increased. It doesn't mean that one caused the other. In another example, the amount of ice cream sold and the number of murders committed both increased during the summer months. It doesn't mean that one caused the other. What we understand is that during the summer months, more people are out, and when they're out, they tend to buy more ice cream and commit more murders. This infographic from the textbook shows that there's a correlation between more coffee and less Parkinson's disease, and less coffee and more Parkinson's disease. But we have to be careful. There are many more explanations besides saying that one causes the other. We need to exercise caution when we look at the results of epidemiological studies. Sometimes it's hard to control for the complexity of a disease. Diseases can manifest themselves differently depending on age, sex, or genetics. We also have to be careful with sample sizes. We want to make sure that the sample size is large enough to give us confidence in the results. After we find a correlation or relationship, we can design a controlled experimental study to understand the link between two variables better. An example of a controlled experimental study is a randomized clinical trial. In a randomized clinical trial, subjects are randomly chosen to be in control in experimental groups to test an independent variable, which is usually an experimental treatment or experimental drug for some condition. If we were studying caffeine, we might have a randomized clinical trial with an experimental group that drank caffeinated coffee and a control group that drank decaffeinated coffee. We'd follow both groups over a number of years to see which group had the higher incidence of a disease like Parkinson's disease. Scientific research and data are often very complex, and the public often only sees this work as isolated media headlines. We need to be critical of these headlines and look at the science behind them if we're going to determine how legitimate they are. These are two tabloid magazine covers that we should evaluate very critically. Before we wrap up this screencast, let's go back and talk about statistical significance. Statistical significance is a measure of how real our results are versus being due to random chance. When we say a result is significant, it means that we are very sure that the result is reliable. In order to determine if a result is statistically significant, we calculate something called the p-value. The p-value is the probability that your result is due to random chance. If the p-value is less than 0.03, it means that there's less than a 3% chance that your result is random. We call this highly statistically significant. If it's greater than 0.03 but less than 0.05, we call this statistically significant. If it's greater than 0.05, we say that the result is not statistically significant. The chance of your result being random is too high to call it significant. At the end of each chapter in your packet, you'll find a summary. Please use the summary to make sure you understand the major ideas in the chapter. In future screencasts, I will leave the summary slides out.